Chapter 13, Experiments and Observational Studies. To date, this is the most important chapter that we've covered. In an observational study, researchers don't assign choices or treatments. They simply observe the subjects. Okay, there's three types. We have retrospective studies. Think retro being in the past. This studies past data. Prospective studies future data. And then cross-sectional studies, think about just taking a slice of something. That studies data from a specific point in time. Okay, when we do observational studies, we can use them to discover trends, but we can't determine causal relationships between two variables. In order to actually identify those causal relationships, we need to conduct a randomized comparative experiment. And in an experiment, it involves applying a treatment to a subject, seeing how it affects one group compared to another. This allows us to prove that cause and effect relationship. There's always at least one explanatory variable, we call it a factor, and at least one response variable that we're measuring. A few other pieces of terminology before we move on to our example. When we're assigning our treatments, the items that receive these treatments are called experimental units, with the exception that if we're assigning our treatments to people, we call them subjects or participants because that's nicer than calling people experimental units. A factor usually has a number of different values it can take on. These are called the levels of the factor. And a treatment is just a combination of all the different levels from all factors within the experiment. Let's use this example to make sure we're clear on all the terminology that we just went over. Here we have a farm products manufacturer who wants to determine if the yield of a crop is different when the soil is treated with 3 different types of fertilizer. We have 15 similar plots of land that are planted with the same type of seed but are fertilized differently. At the end of the growing season, the mean yields from the plots will be compared. Part A, identify the experimental units. So experimental units, aka subjects if we were dealing with people or participants, are what receives the actual treatments. It's saying in the problem that the treatments, okay, quite explicitly, it's treated with fertilizer. So the things that are actually getting this fertilizer are the 15 plots of land. Identify the factors in the experiment and the number of levels of each. So the factors are what are we actually manipulating, what are we changing within our experiment to see how it affects the response variable. So in this case, we just have one factor, it's the fertilizer, at 3 levels. Identify the number of treatment. Okay, since there's just one factor, it's just the levels of that factor. There's 3 different treatments. And the response variable. What are we actually measuring at the end of this experiment? We're going to measure the mean yields from this crop across the different plots. Now that we've covered the terminology, let's go through the 4 principles of experimental design. Now, the first is control. And what we're talking about when we're talking about control within an experiment is minimizing any external sources of variation or any external influences that might affect our experiment. To note, in that last example, it had mentioned that all of those 15 plots of land that were receiving the fertilizer all had been planted with the same seed. So they didn't have tomatoes on some, corn on another, peppers on another. Okay, we make all of our conditions, all of our plots of land as similar as possible so that any of the differences that we see can be attributed directly to the fertilizer that they receive. Okay, some other things that we might want to control in that experiment is the amount of water that each plot gets. If there's any pests, make sure that they're all getting the same pesticides. If we can control how much sunlight that they get, all those things that might affect the growth of the plants on these plots would want to be controlled as much as we possibly can. Obviously, we can't control everything, which is where this next principle comes in, the principle of randomization. So sometimes we're unable to control for certain influences. Sometimes we don't even think to control for certain variables that might end up affecting our experiment. So what we do to deal with all of those at the same time is we randomize which subjects, which experimental units get which treatment. Okay, and this randomization spreads out all of these different external sources of variation and it equalizes them. It doesn't eliminate the effects of these sources, but if we're randomizing which plots get which fertilizers, it should spread them out to minimize the effect that it has on a response variable. Principle number 3 is replication. Think of it as repetition or the fact that we want to duplicate this process as many times as we can. Throughout the entire course, more data is always better as long as it's collected appropriately. Within this example, we have 15 plots of land, we have 3 fertilizers, so we'll be duplicating this experiment in a sense by giving 5 different plots each of these 3 different types of fertilizers. If one of these fertilizers actually is better than the others, we should see that even if there is some kind of external source of variation, like maybe one of these plots gets a little less water or a little less sunlight. Lastly, our 4th principle is blocking. This is not essential. It's not going to be used in every experiment, but we do want to use it where it's required. And it's required anytime we have a variable which we expect would have an effect on a response variable. For example, on the bottom here, we have what's called a design diagram. And it's showing an experiment where we have 3 different treatments. Okay, but before giving these treatments, the examiners decided that males and females might have different responses to these treatments. So before they did anything else, they split people up by their sex. Okay, once we had men and women separated, that's when we randomly assigned them the 3 treatments. Okay, we gave it to women, we randomly assigned them separately to males. Okay, and then we run our experiment. That allows us to get one set of results for the women, one set of the results for the males. We can see if there's any differences between the 2 sexes. We can see differences between the treatments. Okay, so we do that because we think there might be a difference in the responses to those 2 genders. It's a lot like stratified sampling. It's really almost exactly the same, with the only difference being that we stratify in samples and we block in experiments. Tying this back to the example that we've been doing, let's say that in our 15 plots of land, they were arranged in 5 rows of 3 like this. And let's say at the very top of our plot of land, we had a river. This is water. Okay, and then down at the bottom here, there was a road. So we might want to block where all 3 of these plots right here get one of those 2 types of treatments, one of those 3 fertilizers, and same thing with every one of those rows until we get to the road. What's important is that we still include randomization, so we would randomly decide which of the 3 boxes gets which fertilizer on every single row. Control groups. It's important that every experiment has some kind of comparative element so we can actually compare the results of our different groups. It's good to have a baseline group to compare our results against, and that baseline group is called a control group. Control group can have no treatment, or it can just have a treatment with a known result. Maybe we're testing a new migraine medication versus a migraine medication that's been around for a really long time. Or on the opposite side of the coin, maybe we're testing a new migraine medication versus a placebo, where someone's getting a pill that actually has no medicinal properties whatsoever. And with that lead in, let's talk about placebos along with the overall concept of blinding within an experiment. Many times in an experiment, we won't want our subjects to know which treatment they received. This is called blinding. And blinding can occur at 2 different levels. We can blind the people who are being treated, the subjects being treated, or we can blind the people who are evaluating those subjects. Okay, sometimes we'll actually blind both of those groups. If we're blinding one of those 2 groups, the experiment is said to be single blind. If everyone in both of those groups is blind to the treatments, this is called a double blind experiment. I had mentioned a placebo on the previous slide. A placebo is just a fake treatment. It looks just like the real treatment. Okay, it's a method of blinding, and it's meant to kind of trick our subjects or our evaluators into not knowing which treatment was received. So after we've done our experiment and collected our data, we're almost always going to have differences between our treatment groups. This is natural. Variation is natural. We should expect to see those differences. What we're really looking for are differences which are statistically significant. Okay, we're going to learn more about what that means precisely as we move a little bit further into the course. But for now, statistically significant differences are just differences that are larger than we'd get from chance alone. Just a few last things to close out this set of notes. In observational studies, sometimes we'll pair subjects with others because they're similar in ways that are not under study. This is called matching, and matching reduces variability in the same way that blocking reduces variability in experiments. As an example, let's say we're studying the relationship between music education and grades. So we're going to match each student who studies an instrument with someone of the same sex and family income, but doesn't study an instrument. The goal here is to create or to find two people who are as similar as possible so that the only difference between those two is that one studies an instrument and one does not. Matching is for observational studies. Blocking is for experiments. They both have the same goal, reduce variability within our subjects. Lastly, we've previously talked a lot about lurking variables, especially in Unit 2. A lurking variable creates an association between two variables that implies that one causes the other. Within experimental design problems, we have a different term. It's called confounding. And when variables are confounded, they're affected by two factors at the same time. We can't separate the effects of one factor from the effects of another. Let's say we have our plots of land. We had shown in that diagram, some of them are along a river, the others are along a road. And let's say we didn't randomize our treatments. Okay, so all of the one fertilizer is along the river, all the others along the road, and the third is in the middle there. Unbeknownst to us, there's a factory upstream. They're dumping chemicals into the river. All the plants along the river are really underperforming. They're not growing as well as the other two fertilizers. Is that because that fertilizer is just not as good, or is it because the chemicals in this factory are being absorbed into the soil? We're not sure. There's two factors that are affecting our response variable. This experiment has been confounded. Lurking variables are for observational studies, scatterplots most likely, while confounding variables are for experiments.